Okay, we begin our discussion of this leg of Chapter 3 with a description of the ribosome, and these are the protein builders in the cell. They have a large and a small subunit, so they look a little bit like a set of sandwich buns, if you were to draw a picture of them. And they can either be floating free in the cytoplasm, or they can be attached to the rough endoplasmic reticulum, but their role is the same in either case, which is to manufacture protein. And as you can see in this diagram, when the cell needs a particular protein manufactured, what it'll do is it will send special molecules called transcription factors. Let's draw the transcription factors here into the nuclear core. Okay, so let's say this little green ball is a transcription factor. So it'll move through the nuclear pore, and what'll happen is it'll eventually find the gene that contains the instructions for the protein that the cell requires to be manufactured. The transcription factor, in turn, will guide the copy of the genetic material in that part of the DNA, which is known as a gene, into a molecule of messenger RNA, which will have the exact same sequence as the coding strand of the double-stranded DNA. So this single-stranded message will then be released by RNA polymerase when it gets to the end of the gene and will flow back out through the nuclear pore, at which point the large and small ribosomal subunits will search for the beginning of the message, which we call the 5' end, and then they'll scan that message for what we call the start codon. And the start codon in most eukaryotic genes is AUG. Let's draw that up here. And that first amino acid, as a result, that gets incorporated into the growing peptide chain is a methionine, because AUG codes for methionine. So AUG will just indicate that that's the first codon right there. Now, once the ribosome is assembled onto the messenger RNA and has found the start codon, what it'll do is it'll call for a very important molecule called a transfer RNA, which has sort of a cloverleaf shape. Looks a little bit like this. And attached to this transfer RNA is an amino acid. We'll indicate that in black here. So we'll just call this AA for amino acid. The ribosome will search for the transfer RNA that contains the anticodon that has essentially complementarity to the codon that it's sitting on top of. So in the case of the start codon, AUG, the initiator transfer RNA in this area here called the anticodon loop is going to be UAC. And that first amino acid would be methionine. That's what the genetic code dictates that the ribosome would pull up. The ribosome will then march forward 3 more bases, basically the length of one codon. It'll hold on to this transfer RNA, and it'll bring in now the second transfer RNA bound to the second amino acid that's going to be incorporated into the growing polypeptide chain. And the identity of that amino acid is going to depend on the identity of the codon. So let's just, for fun, say that the next codon was, let's say, UAC. Then the transfer RNA that would be pulled would be one that had the complementary anticodon loop, and that would be AUG. And then that amino acid is attached to that transfer RNA, would be brought into the ribosome, and then the ribosome will do something very interesting, which is it'll create a peptide bond between amino acid number 1 and amino acid number 2 as it moves 3 more bases towards the end of the message, which the end of the message is the 3' end. You can see here. In the beginning of the message up here, this is the 5' end. And this process continues. Once we've moved now this next block of 3 bases, we can release the first transfer RNA because it no longer is attached to an amino acid. That transfer RNA, once it's back in the cytoplasm, will be recharged by enzymes that will attach the appropriate amino acid to the molecule so that it can be reused again at a later point. And this will continue 3 bases at a time, 1 codon at a time, until the ribosome reaches the last codon in the message, which we call a terminator. And the terminator corresponds to a transfer RNA that, instead of being attached to an amino acid, is attached to a water molecule. And what happens at that step is that the water molecule is joined to the terminal amino acid in the polypeptide chain. The polypeptide chain is released, the ribosome will disassociate, and the message may be used again, may be degraded. It all depends on the needs of the cell. Very often, if the product is needed in large amounts, what will happen is that a single message can be read over and over and over again, and that phenomena where you've got many ribosomes moving down a single message is called a polysome. P-O-L-Y, polysome. And there are electron micrographs, I think your book may have one, or you can Google it, that show ribosomes marching down a message, and they also show the nascent polypeptide chain coming out of the ribosome. So basically you've got 2 strands joined together by a ribosome, one strand being the protein, the other strand being the message, and you can actually see protein biosynthesis in action as a result. Okay? And so this continues until when? Until the appropriate amount of protein has been manufactured to meet the needs of the cell. And at that point, what will happen is that the cell will basically send protein commands into the nucleus telling the transcription factor, or inhibiting the transcription factor from opening up that target gene. That gene will be closed up, that message will no longer be produced, and as a result there won't be any more source information to generate that protein in the cytoplasm. So that's one of the ways that we regulate now the the manufacture of protein to meet the cell's needs. Okay, so that's basically the long and the short of it here. Now, one of the things we should also point out about what we're looking at in this slide with regard to ribosomes is how they're assembled. The area of the nucleus that's dedicated to synthesizing the RNA structural component of ribosomes, which is ribosomal RNA, is the nucleolus. It's a dark staining area at the center of the nucleus. And as you can see, in order to assemble a viable ribosome, what has to happen is that the ribosomal proteins, which are actually synthesized in the cytoplasm, are imported into the nucleus, guided to the nucleolus, where they then combine with the ribosomal RNA, and then the entire complex exits through the nuclear pore, where it will rest either on the rough endoplasmic reticulum or will remain a viable unit inside the cytosol. And of course, the purpose of this is to have enough protein assembling machinery to provide for the biosynthesis necessary for the cell to maintain the processes that we associate with growing, reproduction, and repair. The endoplasmic reticulum is essentially where the protein takes on its native shape. The rough endoplasmic reticulum is so deemed because it has attached ribosomes on its surface. The proteins are produced and modified, and they fold up into their native conformation inside the rough ER. The smooth endoplasmic reticulum has no attached ribosomes. It's the site of lipid manufacture, and as a result is essentially providing a lot of the source material for the plasma membrane and the membrane enclosed organelles. And you can see an electron micrograph here of rough endoplasmic reticulum. You can see the ribosome studding the surface. And here you can see in the diagram the smooth endoplasmic reticulum. Um, they're frequently contiguous sets of flattened sacs. What happens as the protein works its way through the rough endoplasmic reticulum is it'll eventually arrive via a vesicle at a structure called the Golgi apparatus. Essentially, as the protein folds into its native shape, it gets boxed up into a little membrane enclosed vesicle, which will fuse with the portion of the Golgi that faces the rough endoplasmic reticulum, which is called the transface. And then what will happen is that particular block of proteins will be labeled inside the Golgi. Essentially, small molecules will be attached to it that have the molecular equivalent of a dress label activity inside the cell. And then those proteins will be boxed up and sent off of the cisphase, and they'll be transported by cytoskeletal proteins to their final destination in the cell, where they'll be dropped off and they'll do their job. Now, these could be protein products that are excreted from the plasma membrane. These could be protein products that are delivered to another membranous organelle. Um, these could be protein products that are actually dropped off within the cytoplasm. It all depends, again, on the function of that particular protein. So you could think of the Golgi apparatus as a distribution center, okay? Very important piece of the endomembrane system, which is essentially a contiguous series of flattened sacs that runs from the nuclear envelope to the rough ER to the Golgi apparatus, from there to the secreting vesicles, and from there ultimately to the delivery of the protein. And what you're looking at here is just an example of how this system works. You can see here the message being bound by the ribosomal subunits and the protein being extruded into the rough endoplasmic reticulum. It folds into its native conformation, and then what happens is that we now bud off of the rough ER when we get essentially to the end of this portion of the endomembrane system, and then we fuse with the Golgi apparatus. You can see here the vesicles fusing, and eventually what will happen is these will be tagged with proteins that will indicate where they're going to be delivered inside the cell so that they can do their job properly. As we move through this series of flattened sacs, proteins with a similar destination end up in the same vesicle, and then those vesicles get guided by cytoskeletal components to their ultimate destination, where they get dropped off. And then one of the things that you'll notice is that these vesicles can also recycle. They can, for instance, if they're exocytotic vesicles, they can deliver their payload to the extracellular environment, and then at some point the cell may decide to recover this little bit of membrane, and as a result these vesicles can move back and forth between the Golgi and their destination, just like trucks leaving a central distribution center can go back and forth between the warehouse and the store where they drop off their inventory. Now, lysosomes are important because what they do is they break down worn out organelles, and they're also involved in recycling components that the cell doesn't want to have to make de novo. You could think of them almost as the stomach of the cell. An example of lysosome action is shown here, where we see endocytosis, which, remember, is a form of active transport. We bring inside the cell something that the cell wants to break down. What happens here is that the lysosome, which contains powerful catalytic enzymes that break down things like proteins and polysaccharides and other macromolecules, will fuse with this vesicle and will break down the components that were brought in via endocytosis, and then components of those breakdown products will be used to manufacture other macromolecules, and then the waste products will eventually be extruded through the cell membrane through processes such as exocytosis. So you can see here that the lysosome is very important because it's one of the ways that we break down worn out stuff, um, and this allows the cell essentially to effectively take out the garbage. There are diseases in which lysosomes are defective, where they're unable to break down waste products inside the cell, and these are called lysosomal storage diseases, and the result very often is that the cell starts to accumulate so much toxic waste that it ends up dying. An example of a lysosomal storage disease is a condition known as Tay-Sachs, in which there's an inability to break down a particular sugar protein molecule called a ganglioside. The result is that it accumulates inside neurons to toxic levels, and it results in the death of neurons. This is generally a lethal condition. Children born with this usually don't make it past infancy. So here's a look at lysosomal action.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. The hydrolytic enzymes degrade proteins, nucleic acids, lipids, and carbohydrates, and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles such as old mitochondria. This results in the destruction and recycling of these structures.
Okay. Peroxisomes are smaller than lysosomes, and they contain enzymes that break down fatty acids and amino acids. Hydrogen peroxide is a byproduct of this breakdown. One of the things that peroxisomes do is that not only do they break down fatty acids and amino acids, they also serve as detoxifiers for the cell because they um, end up destroying molecules that are called free radicals. Free radicals are essentially electron poor species inside living tissue that steal electrons from other macromolecules and cause them to break down. And so the peroxisomes serve as free radical scavengers, resulting now in a minimization of damage to the cell as a result of these free radicals that occasionally crop up during metabolism. Proteasomes are large protein complexes, and they include enzymes that break down and recycle proteins in the cell. One of the things that's important for the cell to maintain integrity is that it doesn't want to have to make everything from scratch. So whenever possible, the cell will try to recycle components and reuse them, and that's part of what the lysosomes and the proteasomes do for the cell, is that um, useful components are retained, and the cell can then incorporate them into macromolecules in the course of biosynthesis. Now, mitochondria are very interesting organelles. They are nicknamed the powerhouse of the cell, and this is where most of the ATP synthesis inside the cell takes place. Two very important metabolic pathways occur in the mitochondria. One is called the Krebs cycle, also known as the citric acid cycle, and the other is the electron transport chain. And essentially, the way these pathways work is that they receive a molecule called pyruvate from glycolysis, which is a metabolic pathway that occurs in the cytoplasm of the cell. And then the pyruvate is slowly oxidized, and as it's oxidized, the chemical potential energy in the pyruvate molecule is transferred to high energy molecules called NADH and FADH2, and there's also some ATP made directly during the course of this process. Eventually what happens is that the NADH and FADH2 are oxidized by the electron transport chain, and that energy is used to generate massive amounts of ATP. These two pathways only run effectively if there is oxygen in the cell. So this is why it's very important that the cell have both oxygen and nutrients delivered by the bloodstream on a continuous basis, because without it, we wouldn't be able to make sufficient ATP to maintain cellular integrity and keep the cell alive. There's two sets of membranes in the mitochondria, an outer and an inner mitochondrial membrane, and as you can see, the inner mitochondrial membrane is highly folded. These little folds and grooves are called cristae, and the components of the electron transport chain actually are embedded in the cristae. The matrix is deep to the cristae. It's sort of like the cytoplasm of the mitochondria, and it contains the enzymes that are responsible for conducting the Krebs cycle, and it also contains mitochondrial DNA. And one of the theories about how the mitochondria came about is that we believe it is a descendant of a very early bacteria. The thinking is that early on in evolution, the first eukaryotic cell formed a symbiotic relationship with one of the early bacteria. The symbiotic relationship essentially guaranteed that if the bacteria could live inside the eukaryotic cell and produce sufficient energy to drive the eukaryotic cell's metabolism as well as the bacterial metabolism, that the eukaryotic cell would provide a safe place for the bacteria to live and also essentially bring the fuel in. So essentially what happened is that the bacteria became so dependent on the eukaryotic cell to exist that they lost their ability to live independently. The reason we think this is what happened is because if we look at mitochondrial DNA and compare it to DNA from organisms that are currently here on the planet, it's very similar to bacterial DNA. So this is a, the theory of how the mitochondria came about. We call that phenomenon the production of an endosymbiont, meaning a symbiotic organism that lives within another organism in order for the relationship to work. The mitochondria increase in number when the cell requires more energy. They also increase in size. And the mitochondrial DNA, of course, codes for proteins that are important for mitochondrial function. Those include the enzymes that are responsible for the Krebs cycle and the electron transport chain. Um, there are also ribosomes inside the mitochondria that engage in protein synthesis. And what's interesting, of course, about those ribosomes is that they're a lot more like bacterial ribosomes than they are eukaryotic ribosomes. So as a result, that's further evidence that indicates essentially that we believe mitochondria descended from bacteria. Next thing we want to touch on are centrioles and spindle fibers. These are located in specialized zones near the nucleus, and what we're looking at here is the structure of an organelle known as the centrosome, which is responsible for the assembly and the breakdown of microtubules. And the significance of microtubules is that they help to maintain cell shape, but they also serve as pulleys and guides to move organelles around inside the cell, and they're critically important in the process known as mitosis, where they attach to duplicated chromosomes, and through the action of the microtubules pulling in opposite directions, they separate the duplicated chromosome into two um, complete separate DNA molecules, one of each that will go to each new cell. Before cell division, the centrioles divide and they move to the ends of the cell and they help organize the spindle fiber. And so if we were to illustrate the role of the centriole, let's draw this would be a chromosome, okay, a single chromosome with this little stricture in the middle. It's called the centromere right here. And then during the synthesis phase, what will happen is that that DNA molecule will be duplicated. So an extra copy will be produced. And these chromosomes will become visible during what we call prophase of mitosis. Okay, so prophase is the first time that we see these duplicated structures. What will happen next is that the 46 chromosomes that make up a typical human cell, that's the total content of the genetic material, will line up at the center of the cell. So again, we're just illustrating here that they become visible during prophase. At metaphase, this is when they line up single file. Okay, so we're just for sake of simplicity showing 3 of the 46 chromosomes. So prophase, chromosomes visible. Metaphase, they line up as indicated here. Okay, and then at anaphase, what happens is that that's when the microtubules are going to attach to the centromeres. So if we draw the microtubules in blue, what happens is that the centriole synthesizes the microtubules, and 2 sets of centrioles move to each end of the cell, and the microtubules and the associated centrioles are what we call the spindle apparatus. So here we have the microtubules attaching to the chromosomes, so you got one at each end. And then these would be, I'll represent these as the centrioles. They're usually 2 barrel-shaped organelles that lie at right angles to each other. And what will happen is that at telophase, here, the centrioles will begin pulling on the microtubules, disassembling them and causing them to shorten. So the microtubules will begin to move in the direction indicated by the black arrows. And what that will result in for the chromosomes is that the chromosomes themselves will pop apart. Okay, so what will happen if we redraw the apparatus over here? And then these would be the chromosomes, and what you'll see is that they're moving to opposite ends of the cell, being pulled by the microtubules, which are being disassembled by the centrioles. Okay, so this represents anaphase, and then telophase is the continued migration of these chromosomes to opposite ends of the cell. Once we reach opposite ends of the cell, what will happen is that the rest of the microtubules will disassemble, the centrioles will become less visible, the nuclear envelope will reform, and each new cell will have a complete copy of the genetic material. Okay, so prophase, chromosomes visible. Metaphase, they line up along the center of the cell approximately. Anaphase is when they split. Okay, so let's do a red arrow showing you anaphase here. This would be anaphase, where they're being pulled apart and split. And then telophase marks the last part of mitosis, where the chromosomes are now at opposite ends of the cell. The microtubules disappear, the nuclear membrane reforms. And then the next thing that will happen after telophase is that we'll move into dividing the rest of the cell contents, and that's a process that's called cytokinesis. Okay, so this leads up here to cytokinesis. We'll just put a CY for cytokinesis. Now, when and where does a cell do this? Well, there are chemical signals that tell the cell when it's appropriate to divide. The first part of that process is triggered when the synthesis phase begins, or S phase in the cell. That's when the DNA is duplicated, and that's really before prophase happens. That's then followed by a small brief period of time when apparently nothing happens, but actually what's occurring is that we're beginning to get ready to make the spindle apparatus and prepare for the first stage of mitosis. So this first gap is called G1. Then we have mitosis, which is prophase, metaphase, anaphase, telophase. Then that's followed by cytokinesis. And then we have another gap, okay, that's called G2. G2 for gap 2. And then we have this, in most cells, a very long period between G2 and synthesis, which we call interphase, or I. Okay, and that's the period during which the cell is performing its normal task inside the human body. And that's essentially the cell cycle. Okay, so the centrioles and the spindle fibers are critical to this very important process. This is what allows growth and repair of tissue. Okay, and we'll get into mitosis a little bit more a little later on in the lecture. Now, cilia are appendages that project from the cell surface that are capable of movement. They convert the chemical potential energy of ATP into kinetic energy, and that allows them either to move material across their surface, or in the case of a related organ known as the flagella, it actually allows the cell to move from point A to point B. And there's only one flagellated cell in the human body, and that's in the male, that's the sperm cell. And so here you can see the power stroke, which involves the moving of the cilia uh, from its resting position to its active position, and then we have the recovery stroke. Okay, so kinetic energy here, recovery there, ATP used in every cycle. Where do we find ciliated cells? One example would be the upper respiratory system, where we have tissue that lines the trachea, which is ciliated pseudostratified columnar epithelium. And what happens in the upper respiratory is it's specialized cells that produce mucus that are called goblet cells generate these secretions, and they lie on top of these ciliated epithelial cells. And as the cilia beat, what happens is that the mucus is moved up towards the oral cavity. The purpose of the mucus is to trap pathogens and debris. The purpose of the cilia moving the mucus up towards the oral cavity is so that you either can spit it out or you can swallow it. In any case, the pathogens and the particulates don't end up in the lungs. Remember that lung tissue is extremely delicate tissue. It's dominated by simple squamous epithelia, and if we were to accumulate particulates and pathogens in the lungs, that would produce irritation, inflammation, and scarring. So that's one of the places where we find cilia. Other places we find cilia are in the male and female reproductive tract, where they help to move gametes through the duct system. And this is what one of the things that facilitates fertilization. Now, flagella are similar to cilia, but they're a bit longer, and there's usually only one per cell. Um, You'll notice both in cilia and flagella that if we were to take a transverse section, what we would see is this 9+2 structure. The 2 here being in the core, and the 9 being these little microtubule components that radiate out. So you can see there 1, 2, 3, 4, 5, 6, 7, 8, 9 total. And what these structures are capable of doing, again, is converting the chemical potential energy in ATP into kinetic energy in the form of movement, so that the flagella is able to produce a whip-like motion, and this is what moves the sperm cell through the male reproductive tract and the female reproductive tract. Essentially, the way that these proteins operate is that um, they're capable of crawling over the top of each other, and that's what produces the curling and whipping motion that we see in the microtubules. Okay, now microvilli are extensions of the plasma membrane that are designed primarily to increase the cell surface area. So we find these in cells that are very important in secretion and absorption. The reason for that is that in cells that have to do a lot of secreting and absorbing, or that have to do a lot of sending and receiving of signals, or um, enzymatic processing of components that are on their surface. We need to do everything we can to provide more space in which to work. And so you could think of microvilli as bench space. Um, they're normally on, there's normally quite a few of them. They tend to dominate the apical sides of the cell, which would be the size of the cell that faces, let's say, the open part of a vessel. And again, what they're allowing is for a lot of biochemical processes to happen in a very small physical space. They don't generally move much. They're about a tenth to a 20th the size of cilia. And where do we find them? An example would be the small intestine. The small intestine is that part of the digestive system that's involved with the secretion of mucus, the enzymatic processing of macronutrients, and also the absorption of the monomeric components of the macronutrients, as well as things like vitamins and minerals and such. And in order to do that effectively, what we need is a lot of surface area. So what you see in the small intestine are really several modifications that increase the surface area. The microvilli being one of them. The microvilli are on the apical surfaces of cells that line the small intestine. So we can draw this kind of here. So these would be the microvilli under a microscope at even at high power. They look like what we call a brush border. Okay, so imagine these as being millions of tiny microvilli, and these cells, these columnar epithelia with villi at their surface, they coat finger-like projections that protrude into the lumen of the small intestine, which are called villi. So what you have are little fingers on top of bigger fingers. Okay, so this big thing here would be a villus. The brush border would be the microvilli. And then beneath the villi are going to be blood vessels. So we'll represent that by a little red line here. And also special extensions of the lymphatic system, which are called lacteals. And we'll draw those in green so you can see those up in here. And what they do is they collect what's absorbed across the lining of the small intestine. The triglycerides, the glycerol fatty acids, they get absorbed into the lacteals, and the monosaccharides and the amino acids get absorbed into the bloodstream. Now, this isn't the only modification that increases surface area in the small intestine. We also have ring-like pleats that line areas such as the duodenum and the jejunum, which are called plicae circulares. So these are ridges, and on top of these ridges would be the villi, and on top of the villi would be the microvilli. And then, as you've probably noticed if you've ever seen a picture of the small intestine from the outside of the body cavity, if we were to say, open up the abdominal pelvic cavity by making a sagittal cut from the xiphoid process, the inferior end of the sternum, all the way down to say where the pubis meets. That's the front part of the hip bone, the os coxa, and we were to open the abdominal pelvic cavity up, and cut through essentially the muscle and the skin, the fat. What we would see if we would look at the small intestine is that it would look like a tube with a lot of twists and turns in it, folded up on itself. Okay, so what you have here are several anatomical modifications that are designed to do one very straightforward thing, which is to increase surface area. Okay, and that's because we have to do a lot of absorbing and secreting and enzymatic processing in a very small physical space. Now, this is just a summary of everything we've talked about up to this point. This is 3.1 in your book. So what I would do is I would star this chart. I would know all the material in it because that'll prepare you for the PRS quiz and also for the exam. Same can be said for, this is just the back end of the table here. Okay, so make sure and understand what's contained in those two tables. Be able to describe it, be able to answer questions about it in a multiple choice or a true-false format. Now, we want to end by talking a little bit about what the genetic material is for, and we've kind of addressed it already when we were discussing ribosomes earlier. This is just a diagram that shows you in a little bit more detail how it is we go from a need that exists in the cell for a particular protein to actually producing that protein in the quantities that the cell needs in order to respond to its environment and maintain life processes. So, to make a long story short, essentially where everything starts is by special proteins called transcription factors going into the nucleus through a nuclear pore, and we'll indicate this here with a yellow arrow. Okay, so transcription factors go in through a nuclear pore, and they look for the region of DNA that contains the information that is going to be used to assemble the protein. Okay, so this would be the transcription factors finding that particular gene. The front of that gene is a gateway sequence that's called the promoter. So it's a little bit like the gatekeeper in a secure community. You have to show ID in order to get inside and visit someone that lives there. The promoter is essentially a gateway to being allowed to read the rest of the gene, and in reading that information, copy it into a molecule called messenger RNA. So let's say that this particular gene has been requested by the cell in order to generate a protein product that's needed. What will happen then is the transcription factors will bind to the promoter, and that region of DNA will unwind so that it becomes readable by a very special protein called RNA polymerase. What will happen is that the RNA polymerase will move from the beginning of the gene, which we call the 5' end, to the end of the gene, which is the 3' end. And as it does that, as it moves down the gene, okay, indicated here by the arrow, it will make a copy of the genetic information in the form of a messenger RNA molecule. Okay. Now, in point of fact, many eukaryotic genes contain pieces of information that don't end up in the final messenger RNA molecule. Those segments are called introns. And so what happens is when the nascent messenger RNA is made, those intervening pieces of information that are not going to contribute to the construction of the protein are spliced out. They're removed. So once the initial RNA molecule has been produced, the introns are spliced out. The 5' end of the molecule is chemically modified. There's a special structure called a cap put on the end in order to indicate, again, where the ribosomes should begin to bind and then scan for what we call the start codon, which would be the first codon in the information that's eventually going to lead to making the protein. So these are a couple of messages that have been made. And then another modification at the 3' end of the message will be that there will be long tracks of adenosines put on the end. That's called the poly A tail. And that's to provide stability for the message. That's also a way to dictate how long the message hangs around once it gets outside the nucleus of the cell. So this messenger RNA, once it's been produced, spliced, capped, and polyadenylated, will leave through a nuclear pore. You can see here, okay? And then the ribosome will look for the cap of the messenger RNA, which indicates the 5' end. And then what it'll do is it'll start walking along the message until it finds the initiation codon, which in most eukaryotic messages is AUG, okay? And it will then incorporate now, it'll stop when it finds that first codon, and then it'll halt when it gets there, and then it'll bring in its first um, transfer RNA coupled to the appropriate amino acid, and you can see those here. The transfer RNAs have a structure called an anticodon. It's the fit between the anticodon and the codon that determines whether or not that particular transfer RNA and its associated amino acid will become incorporated into the growing polypeptide chain. If it is a match, what will happen is that transfer RNA um, will be allowed to participate in the synthesis of the polypeptide. The next thing that will happen is that the ribosome will move down 3 bases to the next codon. It'll incorporate now the next transfer RNA with its attached amino acid. And as we do that, what will happen is 2 things. It'll form a peptide bond between the amino acid that's just been incorporated and the rest of the polypeptide chain. And at the same time, once it moves down to the next codon, it'll release now the transfer RNA that's no longer necessary because its amino acid's already been incorporated in the chain. So it gets pitched. So you can see that process here. What's happening is that the ribosome is reading in this direction. Okay, this would be the start of the message up here. This would be the end of the message. And you can see the transfer RNAs, which are in the cytoplasm, being incorporated by the ribosome as it moves from codon to codon. And that's the growing polypeptide chain. The different colored balls represent different amino acid residues. Now, what will eventually happen is it will reach the last codon in what we call the open reading frame, and at that point, the ribosome, instead of binding to a transfer RNA that's attached to an amino acid, will bind to a transfer RNA that's attached to a water molecule, which is called a termination factor. And when that water molecule is incorporated into the growing polypeptide chain, the polypeptide will be released, the ribosome will disassemble, and the protein will then either fold up into its native conformation in the cytoplasm, if it's a cytoplasmic protein, or it will be extruded into the rough endoplasmic reticulum, where it will eventually fold into its native conformation. When it reaches the end of the rough endoplasmic reticulum, it will bud off into a vesicle. This vesicle will fuse with the Golgi apparatus. The Golgi apparatus will then take that protein and tag it for its final destination in the cell by adding small molecules such as polysaccharides or sometimes lipids or sometimes a combination of both. Those are called post-translational modification. And I should note that this reading of the information in the messenger RNA and using it to make protein is called translation, and the process whereby we make the copy of the DNA and transfer that information into an RNA molecule, that's called transcription. Okay, so transcription comes first, then translation, then the protein folds into its native conformation, then it gets loaded into a vesicle. That vesicle fuses with the Golgi tags the protein with molecules that will end up determining what its final destination is going to be in the cell. And then those proteins will get loaded into another vesicle, which will then be transported to wherever in the cell that protein needs to be dumped off in order to do its job. And all this moving of vesicles, again, happens as a result of cytoskeletal components, such as microtubules and intermediate filaments and microfilaments and so on. And that's essentially how we go from DNA to RNA to protein. That's what we call the central dogma of biology. And we'll get into it in more detail in the next podcast. But what I wanted to talk a little bit about right now is just this idea of simple gene expression. So take a listen to this video, and once that's done, that'll be the end for this block. Make sure and incorporate this material into your notes in preparation for the PRS quiz, and I will see everybody in class. Thank you.
The sequence of nucleotide bases in DNA carries genetic information in units that are called genes. Structural genes encode the information for specific proteins. To create a protein, a gene must first be transcribed into a sequence of nucleotide bases in a messenger RNA molecule. The messenger RNA sequence is then translated into an amino acid sequence of a protein. This sequence of amino acids in a protein molecule determines the shape and chemical characteristics of the protein. Thus, each structural gene specifies a specific protein in the cell that carries out a specific function based on its chemical characteristics and molecular shape. This function of the specific protein gives the cell and the organism the specific trait coded by the gene.